I just wanted to make a quick video introducing the neuron for people who are not familiar with it, just for like intro level biology. So starting off first is the soma. The soma is just another word for cell body. So the soma is right here. It's just a cell body. Just like every other cell you're used to seeing, it has a nucleus right here. So this whole thing is the nucleus. Now, outside of the cell body, we have these little branches right here, right here, right here. Well, these little branches are called dendrites. The dendrites are also sometimes called processes by teachers. A process is just another word for something sticking out of the cell. So even the axon is considered a process. So now you're probably wondering, what's the dendrite used for? Think of the dendrite as the receiving end of the neuron. It receives information from the outside environment and translates it into an electrical impulse in the cell. The dendrites are able to receive information because they have receptors on them which respond to different stimulus. The receptors are, just think of them like much smaller than this square that I drew. They're located inside of these little branches, and they open to little chemical stimulus or mechanical stimulus or voltage stimulus, and they allow for signals to begin getting propagated through the neuron. I'll get into the specifics of what the different channels are, the different channel types are, what the different electrical signals are called, degraded and action potential. That's in a later video. Just continuing forward with the different parts. So once the dendrites create like a little bit of like a signal, like an electrical signal, it starts to move through the cell, right? Starts to move through the cell, and it hits this part right here. This whole part is called the axon hillock. Now the axon hillock is where the electrical impulses from all of these little dendrites, they all get to have stimulus on them, and they all get to provide an electrical impulse, right? And these electrical impulses will get summed right here at the axon hillock. Once there's enough of these electrical impulses, the axon hillock will allow for an action potential to move forward through the neuron all the way to the axon terminals. Now, I'll talk about the action potentials in a different video as well. Just moving forward, we're going to talk about the axon. The axon is all of this here, so axon. In the axon, we have myelin sheath, and the myelin sheath, all it is made up of 20 to 200 layers of modified plasma membrane provided by a supporting cell. So you'll have a supporting cell either in the myelin or on top of the myelin providing the sheath. It's creating the sheath. So the supporting cell can either be oligodendrocytes, as seen in the central nervous system, or it could be Schwann cells. Schwann cells are only in the peripheral nervous system, okay? So there's two different supporting cells for two different nervous systems. Now, moving forward, the myelin sheath, you can think of it as an insulator. It allows the action potential to move down the axon really quickly. If we were going to take out the myelin sheath, the electrical impulses would be considerably slower. Like for people who have multiple sclerosis and Parkinson's, their myelin sheaths have been deteriorated, and it's for that reason their electrical impulses don't move as quickly down the axon, and that's why they have so many issues with propagating signals. So you're probably wondering what these little like little valleys are in between the myelin sheath. These are called nodes of Ranvier, named after the guy Ranvier. So the nodes of Ranvier are found in between the myelin. In the nodes are sodium channels that open in response to a difference in voltage. In my action potential video, I'll talk more about what the sodium pumps actually do. So if you're interested in that, go check out that video. But just know, just to know what it is, just know that the nodes are where voltage gated sodium channels are found, and they allow for the action potential to keep moving, forward, okay? So you can think of it like this. An action potential signal will come, right? I'm going to do it in black, actually. There's a lot of pink here. So it'll come from all this, right? It'll start from the axon hillock. It uses the myelin sheath to jump over. It jumps over all these, like all this space, it just jumps right over it. And then this is where the sodium pumps are. It'll cause another, I'll talk about it later, depolarization, which jumps over this myelin, jumps over this myelin. So that's how myelin just helps this impulse move quicker, because it doesn't have to like walk through these little parts. It can just jump over these whole sections. It's just so much more efficient. It just jumps right over, and then it moves quicker. So the final stop for the action potential is the axon terminal. Axon terminal. Now all of these things right here are axon terminals, right? These little nubs are axon terminals. Now this is where the electrical signal becomes a chemical signal. How is that achieved? Well, because inside of these little nubs, inside of these axon terminals, are vesicles. Vesicles have their own membrane, right? And inside the membrane are little chemical messengers called neurotransmitters, okay? So once the action potential hits these terminus, right? The axon tells these vesicles to open up, and the neurotransmitters flow out into this extracellular space around here, right? So they'll flow out all around here. Now, it doesn't just end there. It's not like they flow out and that's that. They're just floating in extracellular space. No, they're messengers. So what are they targeting? Well, this is where we connect everything we know with the rest of the body, right? So what we do is these vesicles will actually come out and communicate with another neuron. So let's imagine there's another neuron right here, right? Just like a really simple picture of the other neuron, right? Boom, boom, whatever. Here are the little terminus. I guess I'll do just myelin and circles. And then we have the dendrites here, right? Well, this little junction right here between the axon and the dendrite is the synapse. In here, in this synapse, right? So let's just blow it up right here. Inside of this synapse, it looks like this. Where this is the dendrite and this is the axon terminal, right? These little vesicles will move down, move all the way down, and they will eventually merge with the axon terminal, releasing the neurotransmitters into the space right here, right? Into the space between the dendrite and the terminal. So all these neurotransmitters flow out, and then where do they bind? Well, remember when I said that there were receptors on the dendrites that create these electrical impulses? Well, here you go. Inside of this are little receptors. These neurotransmitters will bind to these receptors, cause them to open, and allow ions to flow in, which eventually is the reason why these electrical impulses are created. And once again, this electrical impulse will go down, sum up at the axon hillock, make its way all the way down, jump over the myelin sheaths, right, and hit the axon, where all of this gets repeated again. More vesicles are released, communicates with another neuron, and that's how your central nervous system and your peripheral nervous system is wired. Thanks, guys.